Okay, so we need to understand and remind ourselves of some things, okay, before we start. So let's do some review. So DNA, remember, that lives inside the cell. It's our blueprint. I always call it the recipe book, as you know, because inside it is the genetic material and the instructions then to make all of the proteins that we need to make. And every time a cell divides, the DNA is divided so that every new cell gets an exact copy. The other thing to think about are ribosomes. Ribosomes, remember, are found in the cytoplasm. They're also found on the endoplasmic reticulum, but that's the site of where we build our proteins. I always like to refer to it as the head chef in the kitchen. Excuse me. So protein synthesis is really building proteins, in that we need them because we need to have them in our cells to do jobs, either structural jobs or functional jobs. So either building blocks or proteins that have jobs. Remember we talked about chromatin, and chromatin was the little tiny bits that you find in the nucleus, and it's where the DNA is. And then when we're going to divide the cell, the chromatin all comes together and condenses and creates what we know as chromosomes. So still reviewing, we remember we go back to DNA. We said that DNA is the master blueprint. It's the recipe book. Inside the DNA has segments in it, and each of those segments then has the information to build one protein. So I like to think about it as the information for one recipe, or a page. It's a page in the book, okay? A page in the recipe book. Now there's two other players that we have to think about. RNA, which is a type of nucleic acid, and there's a couple of kinds, but right now we just want to talk about two kinds: messenger RNA and transfer RNA, or commonly called mRNA and tRNA. Protein synthesis then uses both, it requires DNA and RNA. Remember that the gene inside the DNA has the recipe for one protein, and in this information is the sequence of the base pairings, okay? And each of these three bases specifies one amino acid in the protein. I'll show you a picture in a second. Messenger RNA carries the instructions for the protein synthesis from DNA to the ribosome, and transfer RNA then is tasked with going and picking up the various amino acids in the cytoplasm and bringing them back to the ribosome. So there's two steps, okay? Two steps. I think I'll just go to a picture for a minute, okay? All right. So messenger RNA, okay, and transfer RNA. So I just wanted to show you those, what we just talked about. Those three, okay, anticodons, okay, are going to match the message that we have. So hold that thought now, okay? So there are two steps involved: transcription, okay, and translation. So transcription is the information in the DNA, okay? We need to get that information in the DNA, and we can't take it out. DNA doesn't leave. It's like one of those books on reserve at the library. It has to stay there. So we send in messenger RNA, and messenger RNA goes and decodes the information that we need. In other words, messenger RNA goes in and makes a photocopy, okay? So imagine that if you compare, the old VCR tapes. Now I know we don't have those anymore, but it's kind of the best way to imagine it. So we have this recording tape, okay? We can look at that recording tape all we want, but it's not going to tell us anything. We need it to be decoded. So we had a machine called a VCR, and that decoded it for us. I guess it's not unlike a CD, and we put it in the CD player and it decodes us. We can stand and look at the CD all we want, but it's not going to show us the movie there. So we need something to decode it. And that decoding is done by messenger RNA. Remember that we said the manufacturing sites for protein synthesis are the ribosomes. So I like to think of them like a chef in a kitchen, okay? So, because you know, when you see all these kitchen shows on TV, the chef's always shouting orders. So it's the ribosome that says, go get this, go get that, and bring it back to me. So DNA, we're still in the nucleus, however. So DNA is in the nucleus, and it needs a decoder, okay? And we need to get that information back, or we need the messenger to bring it back. So messenger RNA does that. DNA separates. It goes in, and one of its strands is the template for what we need. That's what we need to copy. So messenger RNA goes in and copies it, and then we have a copy of the recipe that we need. The DNA all coils back up again, and now we've got the recipe. So the recipe is brought out to the ribosome, and the ribosome starts telling transfer RNA what the ingredients are. So think of the amino acids like the ingredients, if you will. Okay, and transfer RNA is the sous chef, and it goes and gets the first protein amino acid that it is asked to pick up, and then the second one, and so on, while it reads the recipe. So this definitely needs some pictures. I'm going to come back to this review until we look at some pictures, because this definitely needs pictures. Okay, so I just put some copies of DNA and RNA in there, and so this is trying to demonstrate that DNA has split itself, and RNA is going to take the copy that it needs to take. Okay, so let's look at what's happening here. Okay, so the first thing we did was we said we need a copy of the recipe. Okay, so the recipe is in the DNA, and messenger RNA goes in, and the DNA unfolds itself like you see here. Messenger RNA takes like a copy of it, brings it out, okay, and lays it on the ribosome like you see here, okay, lays it on the ribosome. And so what happens then is, so the ribosome is kind of like a two-piece unit here. So this part goes along and starts to read it, okay? And it reads every 3 of these, okay? And you don't need to know that they're anticodons and codons, but what you need to know is that this represents one particular amino acid, okay? So transfer RNA goes and picks it up. See here, picks it up, brings it back, and attaches it. And we keep making this little ball, this little chain like this. These are all different amino acids that have been read down here. And so I think it's kind of like those Fisher-Price baby beads that babies, you know, you pop together for babies. So imagine the first 3, okay? The first thing is this blue ball. That's the first part. The second one is the brown ball. The second amino acid is the yellow, and so on, until we get this big long chain. And then when we get to here, the termination order is red, and we have the protein. So this pops off and goes and coils itself up. This is another one. It's numbered. That's why I like it, you know. So numbered, you see number 1, DNA, is copied by messenger RNA. Here's the copy. It's going out to the ribosome. The ribosome starts to read it. Transfer RNA is tasked with picking it up and attaching them, and then we have the long block. You know what? All these names here, don't worry. They just represent, the sequence represents a certain amino acid along the way. And the instructions are different for everyone. If you're going to be a research scientist, you'd have to know that. But right now, all we need to know is the concept of it. So with that in mind, I just want to go back to the review here. Okay, messenger RNA makes a copy of on the DNA template. So while I'm doing this, you probably should take a look at the pictures. Messenger RNA leaves the nucleus and goes to the ribosome, and we start to translate it. So it's transcription versus translation. The amino acids that we stored in the cytoplasm are picked up as the ribosome directs transfer RNA to go and get. So transfer RNA goes and gets exactly what it's asked for. They start attaching it. Okay, the ribosome continues to read the recipe, and eventually the termination order is read, and that long chain of balls gets released, and we have our amino acid. So, you know, I talked to different students, and some of them say, oh, I learned this once and the teacher told us it was like building a canoe. So as long as you get a concept in your head and it works, that's the most important thing. I think being able to imagine it really helps you understand it. So lastly there, just to finish up this unit, recognize the significance of DNA. The most important thing is that we need to, it's the recipe, we need to pass it on to each new cell so they get the copy. Just showing you some replication pictures there, and finally how it all comes together. So that's the end of cellular metabolism. The next section we're going to look at is tissues, I believe. Okay,